So, hold on. Good. Okay, so it's recording right here. So here we go. Same question. You listening? Right there. What serous membrane am I touching? Visceral peritoneum, right? And what serous membrane am I touching right here? Parietal peritoneum. Good. And then what is the space between the visceral and the parietal peritoneum? Peritoneal cavity. What's in the peritoneal cavity on a good day? Just serous fluid, right? Just serous fluid, right? On a bad day, it could have any number of things. Okay, everybody cool with that? You all right? Okay, so the one thing that we didn't talk about last time was that some organs are actually not wrapped in visceral peritoneum. So remember the like cookie metaphor that I gave you guys last time? So what I want you to imagine is, of course, we're putting our cookie in the Pyrex dish and we're using crystal pink saran wrap because it's a very special cookie going to a very special person. And we're wrapping the dish, and then we're taking the same sheet, and then we're wrapping the cookie, and then we're putting it in there. And there's 2 layers, yeah, between the cookie and the Pyrex dish. But some organs were in the dish before you started wrapping. Do you see that right there? And that organ is considered retroperitoneal, meaning it was in there before you started putting down the parietal peritoneum. So it ends up being retro, which means what? Behind the peritoneum. Yes? Retroperitoneal. And organs that are retroperitoneal, do you imagine that they will flop around in the body cavity more or less than the other ones? Less, yes. They're smashed back there, kind of like packing tape back there. The kidneys are retroperitoneal. The pancreas and a few other organs are retroperitoneal. So do you see that that's different? Because this one, do you see how the liver has a complete wrapping all the way around it before it hits the body wall and you have 2 layers? Do you see how this one doesn't have 2 layers? So that concept is called retroperitoneal. That's what that is, okay? Organs that are retroperitoneal tend to be pretty stationary and smash back against the body wall. I don't know if you guys know this, but if you were, for instance, going to have a kidney surgery, where do they usually approach it from? Anterior or posterior? Posterior, yeah. They're smashed back behind the body wall. They're really far back there. Okay, so that's the concept of retroperitoneal. And we already did pleurisy and pericarditis, right? But we didn't do peritonitis, did we? Last time? Okay, so let's just briefly talk about what peritonitis is. So basically all 3 of these disorders are when you irritate the serous membrane. And if you irritate it, it either does one of 2 things. It either starts to overproduce fluid or underproduce fluid. We know that it overproduces fluid here, and it reduces the pumping and filling activity of the heart, and it can be deadly. And here, it actually does both. In the early stages, it overproduces fluid, which makes it hard to inhale and exhale. In the later stages, it actually underproduces fluid, kind of wear out your pleura. And then your doctor has listened to your lungs before, right? And you put the stethoscope on different portions on your back. If you had pleurisy, and sometimes you can get pleurisy, like just acute pleurisy, because you've had a respiratory ailment. And sometimes it's chronic, like related to smoking or working in a coal mine or whatever. But regardless, in the later stages of, for instance, chronic pleurisy, when you listen, you can hear the visceral and the parietal membranes sliding past one another. So you're listening with the stethoscope and you hear, and eventually they just stick together. They actually form scar tissue adhesions, and it makes it really hard to inhale maximally or exhale maximally in the later stages of pleurisy. Does that make sense? Now, most of us do not get that just from, for instance, having bronchitis. We'll get pleurisy, but it's going to be acute, and it'll go away usually when the bronchitis goes away. But if you have chronic respiratory ailments like asthma that constantly leads to bronchitis, then you can end up with like long-term pleurisy. It's just not the most common way to get it. Smoking is a really good. Did you guys know that smoking was bad for you? Did you know that? Both of my parents are dead because of smoking. I'm from Texas, and one of the things I love about Utah, I do not like that you're always up in my damn business, but I do like that you don't smoke in my damn restaurants. I love that. That's fantastic, right? But also, mind your own business. Okay, not you, just like everyone. Okay, so that's retroperitoneal and those. So peritonitis. Peritonitis is irritation or inflammation of the peritoneum. But since it's all one big sheet, can you imagine that just because you have peritonitis, it doesn't necessarily tell you where the irritation occurred, right? So you could have peritonitis because of appendicitis, but then it doesn't always necessarily feel like it's your appendix, because you could just feel like you have a stomach ache or you have the squirts or you're freaking constipated or whatever. You could have peritonitis because of a reproductive ailment, right? Like your uterus is pissed off, and so it causes peritonitis. Or one of the reasons that ovarian cancer, so where cancer occurs in the ovary is on the superficial surface of the ovary that is just deep to the serous membrane. That's where it generally goes bad. Okay, so what it does is it pisses off your peritoneum, right? And so the problem is that it doesn't always feel like the ovaries. It feels like anywhere, and it gives you the squirts, or it gives you constipation, or it makes you feel like your uterus hurts. Sometimes it's an ovary, and then you don't really know what's going on. And so it doesn't get diagnosed early, and the cancer ends up getting caught in the late stages instead of the early stages, which is why even though this isn't a very fast-growing cancer, it still has a relatively high mortality rate for a slow-growing cancer. And is it because your doctor doesn't know what the heck she's doing? No, it's because just because you have the squirts, you don't do exploratory surgery, right? However, let's go ahead and acknowledge that ovarian cancer has very strong genetic links. Do you know that? So if you have a family history of breast and ovarian cancer, your month-long stomach ache is something that your physician should know, pay attention to, and they should know that you have a family history of breast and ovarian cancer. Because then you can do some kind of imaging and see if things are wonky in there, right? And then if necessary, you might need to do some exploratory surgery, and then you might live through it, because a lot of people do live through ovarian cancer. Okay, so that's peritonitis. Of course, peritonitis is a common complication of any kind of abdominal surgery, because you kind of tick it off when you do that, right? But of course, they know about that, and they can drain it or just wait for it to go down. But anyway, that's peritonitis. All right, so last thing in this set of notes is super easy. So just ways to describe where something occurs. So I want you to imagine that, like, I am the person who does the initial exam, but I'm not the diagnostician, and she's the diagnostician, or you're the diagnostician. And I want you to imagine that, like, I can't actually draw a picture, right? So what I need to do is somehow verbally describe to the diagnostician where the pain is, where the lesion is, where the complaint is, right? I need to tell you, and I need to tell you in a pretty dependable set of terminology. So what I do in that situation is you can either use quadrants, and of course there's only 4 of those, or you can use regions, right? And so I'm going to write down where something occurs. Quadrants are easy because all I do is draw a line from my sternum through my umbilicus between my pubic bones, and then draw a perpendicular line through the umbilicus, and then you end up with 4, right? So for instance, if someone had appendicitis, they might be complaining of a pain in what quadrant? Yeah, right lower quadrant. If I was describing something going on with the liver, it's mostly in right upper quadrant. The stomach is in, the left upper quadrant. And then you might not know what's in here, but you can learn, right? Okay. So all I really need you guys to do with that is find an organ that is really solidly, dependably in that quadrant, right? And be able to tell me what's in there. Should I use small intestine for anything? No, because that's just damn near everywhere, right? Right? So let's use something that actually shows me that you know where you're talking about. Now, since I'm not making you memorize every organ, that tells you what kind of test question it's going to be. It has to be fill in the blank if I'm not making you memorize the same thing, right? So that matters to you. There's some information in the way that I have you guys learn things. So just learn it and make sure that you can defend it. And if it's wishy-washy and you have to go out onto a limb, choose a different one, right? Choose one that doesn't make us have to argue with one another. All right, so now if you want to be more specific, instead of using quadrants, you can use regions. And the regions are more specific, and they use a lot of the prefixes and suffixes that you've been learning. So let's start in the middle. The one that's right here, it's just under the diaphragm, because remember we're in the abdominal cavity now, right? Okay. So it's just under the diaphragm, right smack in the middle. That one's called the epigastric region. What does that mean? What does that word mean? On the stomach. Epi means on or upon the stomach, yeah? Okay. Epigastric, okay? It's actually on the liver too, but the stomach's just right under there. So that's the epigastric region. Let's move down. This one right here is called the umbilical region because the umbilicus is there. And then this one down here is named dome, if you ask me, because that one's called the hypogastric region, which means what? Below the stomach, but it's below everything, right? I would call it the hypointestinal region, but they didn't let me name it, right? So this is the hypogastric region, right? And by the way, it has some overlap with the pubic region, so I have to write a good test question that doesn't like put both of them, because then you won't know which one to put, right? Okay, but this is specifically with the abdominopelvic regions. Okay, and then let's go down either side, doesn't matter which one, right and left. These regions right here are called the hypochondriac regions, but from the original meaning of hypochondriac, not your aunt who thinks she gets dengue fever every time she gets a mosquito bite. Okay, so what are these called? Hypochondriac, why? What does hypochondriac mean? Below the cartilage, because all of this is costal cartilage, yes? That's all costal cartilage that attaches the ribs to the sternum, right? So that's what hypochondriac region, so it's below that cartilage. Go ahead.
Does it bother you that it's hypo and not like anterior chondriac?
No, it's fine. It's below this cartilage, right? I mean, it should be deep too, instead of hypo, but it doesn't bother me. Yeah, so it's it's below this cartilage. It's below, it's actually sort of behind that cartilage, but hypochondriac. So why the heck did people start getting called hypochondriacs? Well, it's because, oh, I got a pain. Oh, I got a pain. Oh, you got some hypochondriac issues. And then eventually you're just hypochondriac. So that's hypochondriac. So now we will move down here. These are called lumbar regions, right and left, simply because that is the lumbar region of my vertebra and spinal cord. Yeah? So that's where your lumbar vertebrae are. And then these guys right down here are called iliac. Why iliac? What's there? Yeah, the ilium is the bone, the iliac crest is the structure, right? So that's iliac. There's another pretty commonly used word for these, and it's called inguinal, and inguinal means groin. And so I put both of them, I think, in your lab manual. I don't care which one you use. They're kind of evenly, commonly used. So iliac or inguinal. So again, if I asked you, name me an organ that's in the right iliac region, what's a safe one? The appendix is a safe one. Name me an organ that's in the right hypochondriac region. The liver. Do you know what's over here? You can't see it, but does anybody know what's over here? Left hypochondriac? The spleen. Yeah, the spleen's over there. You can't necessarily see it, but it's like right smashed back there. So again, find an organ that is safe to put in each one of them, and which is like mostly there, and it'll be a fill-in-the-blank question, and it should be a freebie, right? It should be pretty easy. Cool? Okay, so now, very last thing on this set of notes is just to talk about what happens when it doesn't look like the picture. When it doesn't look like the picture, that is reality, because it is unfortunate, and I realize it's uncomfortable, because you have this entire expensive textbook that's full of images. But all those images really are an idealized version of what might be true in a large percentage of the population. Sometimes a large percentage is only like 40% of the population. So the textbook looks like the textbooks, and the models are really consistent generally, right? But then you go and look at the cadavers, and if there's 12 of them, there might be 4 different iterations of the same structure. So what are you responsible for? Well, you're responsible for knowing anatomic variability and knowing what's the exception and what's the rule. And it's not to trick you. It's because you are going to be dealing with anatomically variable people in the future. So if every single woman in this room stood up here, we are all the same species, right? We all have the same basic physiology, but there's anatomical variability in our external anatomy. Sizes, locations, proportions, all that kind of stuff. They exist internally as well, right? So even if your textbook said that, okay, between this vein and this one, which is the basilic and cephalic vein, there's one vein that runs right here. 30% of the population has 2 that runs right there. And so how do you make yourself successful? Well, if there are 4 models and 4 cadavers in the lab, what do you look at? All of them, right? Because I don't know if you only studied the cadaver, the forearm on this one cadaver, does your lab instructor know that that he or she has to use that one for the lab practical? No, they're going to use whichever one looks like the best way to set up the lab practical. So you really, really have to have a lot of initiative in anatomy lab that you don't necessarily have to have in other labs. Meaning if 2 tables away there's a model that you don't have on your table, you need to get up and go ask them very nicely if they'll swap with you, right, for a little while, right? And if the lab aide or instructor shows you the thigh muscles on one cadaver, you need to move to the next one and see if you can find them on that one. And if you can't, then you ask for help. And you ask for help in ways that show that you have invested in the process. Not teach me this, but I think I know this one, and this one. Are those right? I'm not sure what that one is, right? So you have to be a lot more independent. Otherwise, you're going to miss a lot. You really are. And there's literally no way that somebody can show you everything. So why is it like that? Why can't somebody just tell you all the holes in the damn skull, right? Because no matter how many times I sit with her with a skull and point out every hole in the skull, will she know them if I don't leave and let her find all the holes in the skull? No, I have to leave and let her struggle through it. That's just how it's going to work. I don't let her struggle through it and get it wrong, right? I come back and say, hey, you got that one, high five, you're a rock star, right? Okay, so, but you are going to have to look at every specimen that you can find and every model that you can find every single time. So you're gonna have to get up, you're gonna have to roam around, even if you're an introvert, right? Maybe you can just wait until they're not looking and see, like, I'm gonna talk to you. I just wanna look at this model. And the lab aids are really helpful, I promise they are. Okay, so in order to facilitate the development of good study skills, you're gonna have a quiz over this entire set of notes, plus the prefixes, roots, and suffixes. I'm going to post it later today. I don't know when. Don't email me before midnight because it's still today before midnight. And then you are going to have until Monday at whatever time I decide to do it, okay? And you can go in and do whichever ones you know really quickly, and you can do the other ones later, okay? All right, so, and then I put this at the bottom of some of the sets of notes. This rabbit hole, see? This little guy going down a rabbit hole? Because I love reading about anatomy and physiology. And some of you guys do too, because you're nerds, and I like nerds. And some of you don't. You don't have to read this crap, but National Geographic has a really, really interesting article about the history and use of cadavers, which is pretty cool. And then this one I thought was really interesting because, like I said, the history of cadaver use has been really sketchy. This one has a journal article that investigates the sources of cadavers that are used worldwide, because at this point in history, we should make sure that we are not abusing populations with less financial power, less social power than other populations. So it's actually published where they come from. Read it or don't read it. I never test over this, but I think it's cool, and I'll probably talk about it at a dinner party, and my husband will try to tell me to shut up. And be like, people don't really like to talk about dead people. I'm like, what do people talk about? I don't even know. Maybe that's why I don't hang out with people. Okay, so next thing we're doing is histology, which is what you guys are doing in lab. It's a little difficult when you first start histology to figure out what the hell it has to do with anything or how you're supposed to identify one versus the other, which is why I'm here. Because you need a framework to put this in instead of just looking at pictures and trying to figure out this one's that and this one's that. So the way that I teach histology is that you are going to understand not just what it looks like, but why it looks like that and how to figure out that it's different than another one. Okay, so but before we get started on histology, let's go ahead and acknowledge that ever since you were in 7th grade, somebody taught you what a tissue was. What was it? What's a tissue? What? Oh, what is a tissue? Yeah, it's a group of cells functioning together, right? So you go cell, tissue, organ system, and all that. But that's not a broad enough definition. So let's broaden it out a little more detail because we're in a more detailed class now, right? Okay. So the more detail is that it's not just the cells, it's also the other stuff. So if I were, for instance, a tissue that had one cell next to another, there wouldn't be much room for anything between them, but not all tissues are like that. Some tissues have cell big space before you hit the next cell, and the stuff between the cells is just as important as the cells themselves. Bone, for instance. Bone has cell, big space before you get to another cell, and then what's in between it? It's crystallized calcium and actually collagen fibers that makes it solid. Does that make sense? And that's really important part of the bone, but it's not cells. It's called the extracellular material, also referred to as matrix, yeah? Or you can either have no matrix at all, a bunch of matrix that's hard, or another tissue is entirely different because the matrix is actually made of different stuff. So what about blood, which counterintuitive, but it actually is a tissue. It's a cell, a bunch of space before you get to the other cell, but what's between them is about 90% water, which means that's a fluid tissue instead of a solid one, right? So when you're studying histology at this level, it's not just the cells that matter, it's also the other stuff, which is extracellular material. You don't really have very much of it in epithelial tissues, but once we actually get to connective tissues, you'll have a ton of it. Okay, so you've already learned this in lab. There's 4 basic categories of tissue. Epithelial tissue is what we're going to do today. It is found anywhere you can stick your finger, right? Any surface, right? That's what my anatomy professor always said. Anywhere you can stick your finger. If you could imagine that your finger can be infinitely long and infinitely tiny, like in every pore, epithelial tissue, surface of my eyeball, up my nose, down my digestive tract, and out the other end, right? Up your urethra, ouch, all epithelial tissue, yes? But also it covers internal surfaces. So anytime you come to the boundary of something on an internal surface, like for instance lining a blood vessel, that's going to be epithelial tissue. The superficial surface of a serous membrane should be epithelial tissue, right? Because you're like finding the boundary of something. So everywhere you run into a boundary, like I was an organ and now I'm at the edge of the organ, that's epithelial tissue, right? So you find it covering surfaces, also forming glands. We'll talk about glands later. Connective tissue. Connective tissue is the weird one because you find it like almost everywhere, but never outside the body on a good day, right? So connective tissue is kind of like this. Let me show you a picture. All right, so if you have, for instance, come here. So since this one's on a surface, what's this one? Epithelial tissue. What I always put underneath it is going to be a connective tissue. So it's like the packing material between any tissue and any other tissue. It's going to be connective tissue. And then it also has some other functions. It's also the one that helps you resist gravity. So it's like bone, right? Strapping something to something else, connecting things. Cartilage is also connective tissue. I think connective tissue is probably the hardest one because it's super variable, but I can help you organize it. It's not going to be bad after we get finished with it. And then muscle tissue or muscular tissue either wiggles something somewhere. So basically this one is about movement. So skeletal muscle primarily moves your skeleton, right? Cardiac muscle is exclusive to the heart and moves things through your heart, right? And then all other muscle is smooth muscle. Smooth muscle is found lining any hollow organ that needs to wiggle and isn't the heart, right? Like your urinary bladder, your uterus, your stomach, your intestines, right? And also in the wall of any tube that has to move things. So for instance, like the fallopian tubes or oviducts, right? Which is where you ovulate from your ovary. It has to go down the oviduct, right, into the uterus if it ever wants to be implanted, right? So if it got fertilized, it gets fertilized out here, and then it has to move in. So how do you move it? Smooth muscle contraction in the tube, right? So if there's ever something wiggling and it's not attached to the skeleton or part of your heart, it's probably smooth muscle that's moving it, right? So you find it all over the place. All right. And then the last one is nervous or neural tissue, and that is primarily for control and communication. And of course, where is most of your nervous or neural tissue sequestered? brain and your spinal cord. But, of course, it also has to wire up to other things. So basically, you have most of it sequestered in here. And then you have to wire it to, for instance, the muscle that you're going to contract or the sensory receptor that you need to get information in from. So brain, spinal cord, and then the nerves, which are really just wiring going in and out. Okay, so we are, of course, going to start where you guys started in the lab, which is epithelial tissue, also called epithelia. A singular is an epithelium. And you find this covering body surfaces. And because it's covering body surfaces, it does a lot of surface things. Yeah? So if you're really thick on body surfaces, you're probably going to be for what? Protecting. Yeah? If you're really thin, you might be for transport. Does that make sense? Yeah? Like secretion or absorption, right? Okay? So not all tissues do all things, but you can kind of look at an epithelial tissue, and if it's got 72 layers, it's going to be hard to get things in and out of that. But if it's got one layer and it's super thin, it's probably going to be easy to get things in and out of it. So it is secretion, filtration, absorption, excretion. You also find it lining body cavities, as we mentioned, and then we'll talk about glands later. Okay, let me tell you, before we get to this, I want to tell you one thing about recognizing an epithelial tissue so that you can get this out of the way really quickly. Because epithelial tissues are almost always on surfaces, they have what's called polarity. And polarity just means that one side is different than another. Like when you have a polar molecule in chemistry, it means it's got a negative end and a positive end, right? Okay, so what does that mean when we are talking about epithelial tissues? What it means when you're talking about epithelial tissues is that since epithelial tissues are on surfaces, they have a free surface, meaning nothing is stuck to that side because that's the boundary. And then they've got another surface that has stuff stuck to it. So apical as in apex or top, right? And basal as in bottom. But just so you know, you can turn them upside down and it's still apical and basal, right? And if you know that you're looking for a free surface, it makes it really easy for you to find an epithelial tissue. So you may not have seen this slide before, but can you find the epithelial tissues on this slide? Can you? Right, because they're next to the free surface. So this one's an epithelial tissue. Is this one? No, that's a different tissue. But this one is an epithelial tissue, right next to the free surface. So you look for a free surface, and then whatever's touching that free surface is going to be the epithelial tissue. Got it? So can you find, for instance, on a different slide that you haven't seen before? Let's do this one right here. Can you tell that this is an epithelial tissue and also where it ends? So epithelial tissue is from here to here, and then you see the tissue kind of change pattern. And this is going to be a different kind of tissue. Guess what kind it's going to be? Yeah, it's going to be a connective tissue. Does that make sense? So epithelial tissue has a free surface. So look for that, and that's going to be the what surface of the epithelial tissue? Apical or apical surface, right? And then you're going to move down to where it changes tissue type, which is sometimes easy to see and sometimes not, right? And that's going to be called the basal surface, as in bottom, okay? Even if it's upside down. So let's look at an upside down one. And this is one that you looked at. It's in your textbook. So what surface am I touching here? Apical surface. What surface am I touching here? Basal surface. We'll talk about the basement membrane in just a second, right? So whether it's thin or thick, it's going to have a free surface. So that's called polarity, and all epithelial tissues except glands have what's called polarity. Okay, so let's go back to these, and then I'll come back and tell you about the basement membrane in just a second. Other things that are true about epithelial tissue, even though you can't necessarily see them all the time in an image, but are true conceptually and important to understand, is that epithelial tissues are usually avascular. And even if you've never heard that word before, try to figure out what it means. Without vessels. There's no blood vessels running through them, okay? Meaning that if I did a cross-section of, for instance, muscle, I would see a bunch of blood vessels running through it, but if I do a cross-section of epithelial tissues, I won't. So, and then there's this thing that sounds counterintuitive, so look at number 3. They're avascular, they don't have any blood vessels going through them, but they're really active. They're capable of really rapid cell division and regeneration, which seems counterintuitive, doesn't it? If you're super active, it seems like you would need a lot of blood flow. Well, they do get a lot of blood flow because even the thickest epithelium is microscopically thin. The living portion of it is microscopically thin. And it's sitting on a really, really vascular tissue. And it can get its nutrients and oxygen through diffusion because it's thin enough, and it can diffuse its waste products and carbon dioxide down. Kind of like taking a sandwich. So make a sandwich, pretend you're 7 and nobody's watching you, and you make a sandwich with 3 kinds of jelly, right? Because nobody made you put peanut butter on it, and you're doing your thing, man. So you got 3 kinds of jelly on your sandwich, right? And you accidentally sit it in a puddle on the counter. And you come back to it 30 minutes later. Will the top slice of bread have jelly color on it? Maybe grape. It will, right? Will the puddle be purple? Yes. But not if you did 16 sandwiches, right? Okay. So as long as it's microscopically thin, then diffusion is an effective transport mechanism to get things up and to get things down, right? And all epithelial tissue, even the thick stuff, is microscopically thin. Go ahead.
So when you're saying, like, skin is an easy example, it's sloughing off the top, the new is generating because that's where the new
Yes,
is.
exactly.
The same is the internal epithelial, the same system.
Same structure. And actually, skin is interesting because only the bottom layers of the skin is actually alive. And you don't have to feed the dead ones. So you're actually sloughing off the dead ones. You're feeding the living ones, right? So
inside, it's sloughing off the top.
It depends on whether it's really thick or not, right? With the thick ones, you slough off. Like, for instance, lining the mouth. So let's talk about that. Lining the mouth, all of the cells are living all the way up to the top. But do they exfoliate? Well, yeah, because sometimes you bite the side of your mouth or impale yourself on a Dorito, right? But you know that it's capable of rapid cell division and regeneration because 3 hours after the Dorito incident, do you even remember that it occurred? No, you don't, because of rapid cell division and regeneration and sloughing, so you can replace it. Now, that sounds all well and good, but let's just talk about clinically that there is a negative. Can you think of any negative to the fact that epithelial tissues are capable and predisposed to rapid cell division and regeneration? Obviously, it's really good in wear and tear areas. What was the issue?
More mutations are possible.
More mutations are possible, and if you have mutations and rapid cell division, then you have an increased likelihood of cancer. Yes. So what are the most likely tissues in your body to lose control of the already rapid cell division? Epithelial tissues. So let's think about some of the common cancers. Some of the common cancers are, of course, skin cancer, right? Breast cancer, that's glandular tissue, which is epithelium. Lung cancer, and it occurs in the lining of the lung, that's epithelium. Thyroid cancer, gland, right? Does that make sense? So, do other tissues get cancer? Yes, but statistically, epithelial tissues account for a disproportionate amount because they were already dividing really quickly. And all you have to do is mutate a single cell, and then you can end up with cancer, which brings us to thinking about what you were doing when you were making the poor life decisions that I did when I was super young and decided to go tan. Okay? I know you look cuter when you're tan, right? But then you get these. And I would have looked way cuter if I did not slather myself with baby oil and iodine and lay on the roof like every other redneck child in Texas, right? So what is the problem there? What occurs is you are bashing a mitotic tissue with a known mutagen, which is UV radiation. And then you are just hoping for the best, right? And sometimes the best does not occur. Now, it is even worse when you have inherently light skin because the UV radiation is more likely to make it to darker layers, to the deeper layers. Does that make sense? And the deeper layers are the really rapidly dividing ones. That makes sense? So, do people with inherently dark skin ever get skin cancer? Of course. But who gets the most? People with inherently lighter skin. We are less protected from the sun, right? That makes sense to you guys? And we'll talk about that when we actually do skin, which we actually skip until later so that I can stay with you on muscles where you need me. Okay, so the cells are closely packed in in epithelial tissues, so you have really little or no matrix to speak of. So let's go back to this figure for just a second and say, okay, if the cells are right next to one another, you don't really have room for matrix. So think of epithelial tissue like tile, right? So you got a subfloor, that's the connective tissue, right? And then you put the cells down, but you put them right next to one another so that you make kind of a complete layer, right? And then you don't really have any matrix to speak of. So there really isn't any matrix to worry about. You don't have to label the fibers in it or anything like that because the cells are stuck together. And eventually, next time, we will actually talk about the types of grout that actually exist between the two. We're not going to get to it today, but next time we will. So they're closely packed with little or no matrix. And if you know that, you can start to recognize an epithelial tissue when you see them, right? If you know both that they have a free surface and that they're right next to one another, then you start thinking epithelial tissue. Got it, okay? All right, so let us talk about the basement membrane. Okay, so if we go with our tile metaphor, let's talk about what the basement membrane is with the tile metaphor. So with the tile metaphor, what we have is the subfloor is the connective tissue, right? But what do you put down on top of the subfloor before you start putting the tile? No, not grout. Grout's between. It's the adhesive that you put down first, right? You put down the sticky stuff, right? And then you put down the tiles, and then you grout last. So we'll talk about the grout next. You with me so far? So it's the adhesive that you put down. And so what the adhesive is going to be is basically it's going to be secreted from both the connective tissue and the epithelial tissue. And when it comes together, it's a bunch of proteins and sugars. It's sticky stuff that keeps the tile stuck to the subfloor. And that sticky stuff is actually the basement membrane, which is sometimes really visible and sometimes it isn't, but it has to be there or else the damn thing would fall off, right? Okay, so the basement membrane is a structure. It's not made of cells, it's made of secretions of cells, from cells, right? And so let's figure out the relationship. You guys tell me because people get this confused. So if I am going from the apical surface to the basement membrane, I want you to put these two words together, two phrases. The basal surface is blank to the basement membrane. The basal surface is blank to the basement membrane. It's superficial to the basement membrane, yes? And the basement membrane is deep to the basal surface of the epithelial tissue. Does that make sense? So are they interchangeable? No. But do I have to be careful when I write a question so that you'll answer it the right way? Yeah, if I ask for a surface, answer basal surface. But if I ask for a structure, yeah, you answer basement membrane, right? And I always consider it my job to write a clear question that would pull the correct answer out of a person that knew it, right? Does that make sense? So you've got the basement membrane, and that's the sticky stuff. The only other thing is that to make really, really sure that this stays stuck. So in addition to the basement membrane, which is the sticky stuff, you got the tiles on there. We'll talk about the grout later. But I want you to imagine that I really, really want this whole layer to stay stuck. So let's say every fifth cell, I pop a rivet all the way through the cell into the subfloor. And that is called a hemidesmosome. And a hemidesmosome, I'll show you what they look like. A hemidesmosome makes sure that the thing stays stuck. Can you see it right here, this hemidesmosome? It's actually attaching, okay? So what it really does is it actually attaches with some protein fibers from the cytoskeleton of the epithelial tissue into the fibers of the connective tissue and keeps it stuck. And you've actually experienced hemidesmosomes before, but you might not have noticed. Okay, so really common misconception is that you think that when you soak in the tub for too long, your fingers shrink, but they don't shrink. They actually swell. And why would they swell? Well, you're sitting in a hypotonic solution, so they sure as hell shouldn't shrink. What should they do if you're sitting in a hypotonic solution? They should swell, right? But what happens is they swell, but they swell up wherever you don't have a hemidesmosome. They stay stuck wherever you do have a hemidesmosome, yes? And so they end up being wrinkly. But if you had a super, super sensitive scale and you weighed yourself after you soaked in the tub for an hour because you freaking deserve it, don't let anybody tell you you don't, okay, and have wine and make the kids go away, okay? Soak in there. You would actually weigh more. You weigh slightly more after you've soaked in the tub because you're actually swelling, but the hemidesmosomes are keeping the spot wells down over there, which is really cool to think about it. You can see it best in your hands and your feet. Does anybody else have kids? My son was terrified of that. My son was terrified. Every time he took a bath, he would get out and his fingers would be all wrinkly and he would totally think they were stuck that way. And so there was just like so many tears. And that child literally never believes me about anything related to anatomy and physiology. He always asks his dad. His dad's a freaking programmer with a philosophy degree. What the hell does he know about anything? But he's like, Dad, is it going to go back? And he's like, your mom says it was, but Dad! And then he also insists still to this day at 13 that blood is blue. I'm like, damn it, do I have to cut you? To show you? He insists. I think at this point he's just doing it to bug me, but before it was real. All right. So you should be able to now, you don't have to do it at this moment, but isn't it easy for you at this point to label apical surface, basal surface, basement membrane, and even though I didn't ask you to, you could probably tell me what kind of tissue that is. Yeah? All right. So cool. Now what we are going to do is getting into the specific tissues, right, which is what you guys did in lab. All right. So epithelial tissues are not that hard after you get used to them because at the very least there's an easy way to recognize them because they always have a free surface, right? All the cells are crowded close together. And then they have a naming convention that's relatively consistent. There are exceptions because literally there is not a biology thing that is a rule without an exception. But generally they have this three-name naming convention in which the first word of the three-name name, like simple squamous epithelium, the first word relates to how many layers of cells there are between the basement membrane and the apical surface. If it's one, you call it simple. If it's more than one, you call it stratified, right? If it looks like more than one, but it's fibbing, you call it pseudostratified, and I'll show you those last. Cool? Okay, so the first word is how many layers do you have between the basement membrane and the apical surface. And then the second word is if I look at an individual cell, what's its shape? If it's squashed flat, right, like a fried egg, it's squamous. Squamous, squashed, okay? Usually with squamous cells, the nucleus is squished laterally. So it's like looking at a fried egg from the side. Good? If it's a cuboidal cell, it's usually about as wide as it is tall, and usually the nucleus is nice and round. I think this is a really cute tissue. I don't know, it looks happy. Cuboidal, okay? And then if it's columnar, it's taller than it is wide. Usually, not always, the nucleus is oval. Now, the reason I say usually is because you never want to memorize a single criterion to identify a tissue, but sometimes if you're like having a hard time, that little extra one can help you, okay? So basically, usually you end up with this three-name naming system in which it's stratified, columnar, epithelium, simple, cuboidal, epithelium, okay? Every once in a while, though, there'll be modifications, like it's going to be ciliated or something like that, and we'll talk about those a little bit later, but just start with the three-name naming system. Okay, so let's start meeting the simple tissues, and the simple tissues are literally the simplest to recognize, okay? So you have all of three are simple, so how many layers of cells are you going to run into? One. Okay, 3 different shapes, and they're all epithelium. So do not get into this bad habit. Some people, I will show them a slide, and they will say simple cuboidal. But you just did 2 adjectives without a noun. So simple cuboidal what? Kumquat, elephant? You need a noun. What's the noun? Epithelium, right? So it is simple cuboidal epithelium. So let's do the simple tissues. So the first one is, let's look at this figure. This figure actually has 2 simple tissues on it. Only one of them is labeled though. Yeah, it's got 2 simple tissues. So what's this one? Simple squamous epithelium, exactly. Now, simple squamous epithelium, for me, I will always ask it from a side view like this. Even though in your lab book, I argued, but even though in the lab, actually not in the book because I wrote the book, but in the the flow chart and in the images, they show a top view of simple squamous epithelium. Here's my problem with that. If you look at a top view, yes, you can see a lot more, but can you tell it's simple from a top view? Can you tell it's squamous? No, so you're just memorizing a picture, right? Which to me doesn't seem really meaningful. If you can recognize simple squamous epithelium from a side view, regardless of where it is, then you actually know what it is. And you also probably know that, do you think this tissue is going to be super duper good at protection? No, but it's boss at transport, right? It provides no impediment whatsoever, really, to transport. So can you recognize it if you see it here? Yeah? You've got a free surface, you've got a really, really thin, squash flat nucleus, and then you change tissue types, right? So that's simple squamous epithelium. All right, but this, as I mentioned, actually has two tissue types. What's this one? Simple cuboidal epithelium. Exactly, that's simple cuboidal epithelium. Good? Okay, so regardless of how you see it, you can recognize simple cuboidal epithelium. So let's look at it here. Again, isn't it kind of a cute tissue? I just think it's kind of cute. It looks happy, right? It's like a nice round nucleus, usually nice round nucleus. One layer of cells that are about as wide as they are tall, okay? And then here's one. Can you recognize that this is simple cuboidal epithelium even though it's a different kind of section? Yeah, you can, right? Nice round nuclei, about as wide as it is tall, right, on the surface, right? And so it starts getting really easy after you've looked at a few of them. And then it doesn't really matter whether it's a longitudinal section of a tube or a cross section of the same tube, right? Does that make sense? And you get good at what? Yeah, okay, so let's go back and talk about this. So one of the things that we will do as we get through going with these, as we are going through all of these tissues, is that both for lecture and for lab, but especially for lecture, don't memorize a tissue without being able to put it into the context of the human body. So let's talk a little bit about where you find simple squamous epithelium. Well, you're not going to find it on the surface of the skin because that wouldn't be a logical place to put a super, super thin tissue. You're going to find it in places that really, really want to do a lot of transport. So you will find it forming the oxygen transport portions of your lungs called the alveoli of your lungs. You will also find it forming the walls of capillaries, because what are capillaries really trying to do? Let oxygen and nutrients out, like waste and carbon dioxide in. Does that make sense? So, and then let's go to simple cuboidal epithelium. Now, I realize that you guys probably don't know a lot about kidneys yet, but let me tell you that you find, so just stick with me and you're going to learn more about kidneys later. Your kidneys do a lot of transport, but primarily the transport's moving out, right? Okay, so what I am going to have in the kidneys is a whole bunch of tubes that contain pee, right? Next to a whole bunch of tubes, capillaries, that contain blood, right? And what I need to do is dump the trash, keep the good stuff, right? And so I got a whole bunch of these. They're called kidney tubules, right? And what they do is really quick, efficient transport at the kidney. And you'll learn a lot about them later in the semester, okay? So you find those in the kidney tubules, and primarily what they're doing is stuff out, right? Although you do pull stuff from the urine. Like you don't want to pee out glucose if you could avoid it, because that's like money, right? You want to keep it if you can avoid it. Okay, if you can keep it. Okay, so now the last one for the simple tissues is there's two different types of simple columnar epithelium, but before we like separate the types, let's make sure that we can recognize it as simple columnar. Okay, so with simple columnar epithelium, let's look at a couple of figures of it. Okay, so when you look at simple columnar epithelium, this is the figure from your textbook, so let's look at this one first. So can you recognize first that it's an epithelium? Yeah? And then second that it's simple, right? And don't get confused because it looks like there's a whole bunch of nuclei down there. What matters is that if there is more than one layer of nuclei, right? So these are all pretty much in the same layer. You with me? Okay. And are they taller than they are wide? Yeah? And are most of the nuclei look oval, right? So that's a simple columnar epithelium. Good? So let's look at another simple columnar epithelium that wasn't from your textbook and see if you can figure out that that one is simple columnar. Yeah? Look right here especially. right? So by the way, sometimes you'll have confusing areas where like, and why is it confusing? Well, because this one is undergoing cell division. So here's what I will do for you. When I make a quiz for you, pay attention to the part that I'm pointing you to, because that's the part I think will not mislead you, right? I am not trying to make you flunk anatomy. People flunk anatomy on their own. They do not need my help, right? So if I point you to an area, focus on that area. So can you tell from here that this is a simple columnar epithelium? All right, so now let's look at a couple of other ones. Now these are a little different, and we'll get to the differences in just a second, but focus on the similarities first, especially if I point to this area right here. Can you tell that this is a simple columnar epithelium? Yeah? Good. All right. So now with simple columnar epithelia, there's two different categories. There's straight simple columnar epithelium that doesn't have an extra word, and then there's ciliated simple columnar epithelium. So now is the time for me to teach you the difference between the two of those. So let me show you the difference. Both of these tissues are found in areas where you want to either do transport, but which one do you think is found in an area where you also want to kind of wiggle stuff? The ciliated. So let me show you the difference between cilia and microvilli now, and then we'll revisit it a little bit later. So let's start with this one. Okay, so if they're both simple columnar epithelia, and they are, I'm going to have two cells that are both simple and columnar, right? Good. Okay. But this one's going to be different than that one. This one, actually, the top side of the cell has a highly folded cell membrane that kind of looks like Bart Simpson's hair. See that, right? Okay. Highly folded cell membrane that looks like Bart Simpson's hair. And this one looks like a minion. Okay. One of these has just massively increased the surface area and can therefore increase transport rate. Which one? A or B? Yeah. These are called microvilli, and they don't wiggle. They're just increasing surface area, because if I need to bring a whole bunch of things in and out, then let's just find an area where it's easy to do that. Let me show you what that looks like. Hold on, let me find that figure for you. I didn't get this far in the other class. Just a second, I'll find it for you. There it is. Okay. So if you looked at one of those cells up really close, they would actually look like that. Do you see that? Okay. And if that all had, for instance, transport proteins like carriers and channels on it, then you could move a whole bunch more, right? So that's microvilli. This one has instead a bunch of little protein projections sticking off with mitochondria at the bottom. It makes it kind of like an outward motor, and it moves on the top, right? But it's not changing the surface area. So these are called cilia. Okay, now I'm going to tell you two different locations so you can figure out the logic of these two things. Okay. I want you to tell me which one, A or B, is located lining the fallopian tubes. Cilia. Why? It has to move the egg. So the combination of smooth muscle in the wall and the cilia lining it moves the egg down. Okay? And I want you to tell me now, you already know the answer, but now I want you to explain the logic of why B is found lining the small intestine. Absorb nutrients way faster. Does that make sense? So they're two different structures that are both found on simple squamous epithelium. And now let's see how we can identify them from one another, because it's really not that hard after you get used to looking at it. What? Yeah, sorry, simple columnar epithelium. So what do microvilli look like when you're looking at it kind of far away like this? So listen. Microvilli look like somebody took a highlighter and just highlighted the surface, right? It doesn't look like bedhead. It doesn't look like hairs. It looks like somebody took a pink highlighter and highlighted the top of it. Do you see that? Now, contrast that with this. Do you see some bedhead going on there? Right? you can totally tell the difference between cilia and microvilli after you have looked at 3 or 4, right? And now, if you really understand that, then instead of me saying, are these cilia or microvilli, eventually I can ask you, is this tissue found lining the fallopian tubes, or is it found lining the small intestine? Right, and then you put structure function together, and then histology ceases to be unrelated to anything, right? Which is how I want you to approach it. And although you won't like it, every single body system that we do after this, I revisit the histology that you learned in the beginning of the semester, and you're like, oh my God, shut up about histology, but I literally never will. Because otherwise you're going to do a brain dump. You'd be like, that was a terrible time in my life, and I don't want to remember it, right? So I just keep bringing up your histology trauma, because I'm a jerk. Okay, so can you tell the difference between those two? Okay, so simple columnar epithelium, ciliated simple columnar epithelium. The only way that you distinguish them in words is by adding the word ciliated. This one doesn't, it's not called microvillide, it's just called simple columnar epithelium. Everybody okay with that?
So if it's not ciliated, it has microvilli?
Well, actually, kinda. Let's just be honest. You actually don't learn every tissue that exists in the body, right? But I promise you that if I show you a tissue and you can't see cilia, just call it simple columnar epithelium, because there are actually more than these two, right? So why are you not learning every tissue in the body? Well, because you're already really busy, right? So there are some that look slightly different than this. So from this, you can, of course, see all the simple epithelia with different shapes here, right? And although Melanie will never ask you from that view, they're probably trying to represent what with that? Simple squamous epithelium. But this is a better way, because that one is simple squamous epithelium. What's this one? Simple cuboidal. And this kind of addresses his question. Are there any simple columnar epithelia without microvilli? There totally are, but I don't nitpick you about it. So if you don't see cilia, just call it simple columnar epithelium. I think this one's trying to represent cilia, but it's kind of a crappy picture to tell the difference. When my picture on the board is better, you know that it's a crappy picture because I can't draw it all. Okay. Now, briefly, I'm not going to get into this very much. When you get into the stratified epithelial tissues, basically what you're going to run into is anything more than one layer from the superficial from the basal surface, basement membrane to the apical surface. Anything more than one is going to be a stratified tissue. So even though you're not going to label them specifically right now, and we'll do it next time, can you tell that this is a stratified tissue? Right? So it's epithelium, right? But there's way more than one layer of nuclei going down, right? And specifically, the nuclei are really the indicators of the cell, although you don't see them in all cells because you just didn't get them sectioned in all cells, okay? So likewise, in this figure, can you tell it's stratified, right? And later on, you will be able to tell what kind of stratified it is. Now, let's do something entirely different. Can you tell that this is first an epithelium? Yes, because it's got a free surface, right? And then now see if you can label it for me, even though you've never seen this figure before. Stratified, cuboidal, epithelium, exactly. And what is all this business? Connective tissue, right? So you're starting to get it. So let's do just one more. This one's a little harder. Can you tell if you look right here? That one's stratified cuboidal epithelium as well, right? And stratified columnar, I'll show you guys next time. It's not very common. It really primarily exists in the distal 1/3 of the male urethra, not the female urethra. The female urethra is really short, right? But the male urethra has to go all the way from urinary bladder to the tip of the penis. So the male urethra is on average 8 inches long, not the penis, ladies. Do not have those kinds of expectations, but all the way from the urinary bladder on the inside to the tip of the penis, and then in the last 1/3, it changes tissue type for reasons we'll discuss in the urinary and the reproductive system. So we will stop there for today, and I will not see you for a week. So I will post your quiz at some point today, as soon as I get a chance to. Okay?